titled Molecular Diagnosis in Clinical Parasitology, a Game-Changing Approach. First of all, I would like to remind to all participants to switch off your microphone and video during the presentation. You can switch back the microphone and video later if you have any questions during the Q&A session. Yeah. So today it's a great pleasure for me to introduce my dear friend, the speaker, Yang Mulia Associate Professor Technologist, Dr. Tengku Shahrul Anuar Tengku Ahmad Basri, in one breath. Or in short, I call him Dr. Tengku lah. Okay. Dr. Tengku obtained his PhD degree in Parasitology from the Faculty of Medicine, University Kebangsaan Malaysia in 2013. His research focuses on neglected tropical diseases, primarily among the marginalized population, utilizing advanced tools to understand the diseases as well as their route of transmission. He started his career as a senior lecturer at the Faculty of Health Sciences, UiTM Puncak Alam campus, and in 2016, he joined I Promise as a research fellow. He is also a former visiting fellow at the Universitas Airlangga, Indonesia. At present, he is an associate editor for the Journal of PMC Infectious Diseases, member of the Malaysian Society of Parasitology and Tropical Medicine, member of International Society of Infectious Diseases, and a professional technologist registered with MBOT. All right, Dr. Tengku, the floor is yours. Game on.
Thank you. All right, let me share the slide first, yeah? All right, Bismillahirrahmanirrahim. Assalamualaikum, everyone. Hello and good morning. Hope all of you are in good condition as well as in a good health. First of all, thank you so much, technologist Dr. Richard Johari James, for that warm and kind introduction. And before I go any further, I would like to take this opportunity to thank the Office of Deputy Vice Chancellor, Research and Innovation for this great and tremendous opportunity. And also, not to forget, to all the viewers who are currently joining me for this webinar session, whether you are actually watching me through this Google Meet, FB Live, or even live streaming. Muchas gracias. So for the next 60 minutes, I will try my best, to share some of our lab experience and thought in using this molecular method to rapidly detect and identify parasite from human samples, and how we could actually use this approach as a part of practicing in our routine lab diagnostic. I personally believe that this topic is timely relevant, as most of us have started to aware, alert, and make ourselves familiarized with the terminology such as PCR, sensitivity, and specificity. And all these words have been frequently used by our Director General of Health during his press con due to COVID-19. So my dear fellow friend, these are the main topic which will be covered and discussed throughout this session. So let's get started. Have you ever come across with this movie advertisement? Actually, it is a film by Bong Joon-ho. I'm pretty sure that some of you might even watch this movie in the cinema last year, or through Netflix during the MCO period. And for your general information, a little bit, this movie has won 4 Oscar awards somewhere in February earlier of this year. And basically, I'm sitting here is not trying to promote or do review about this movie, but what actually catch my attention is the title itself, Parasite. So, what is parasite? And according to the Oxford Dictionary, parasite is an organism who live on or organism that live in another organism, which usually being referred as a host, peruma. And this parasite can be further divided into 2 different groups. First, we have ectoparasite, which is a parasite who live on the external surface of the host. For example, lice. And on the other hand, we also have the endoparasite. So what is endoparasite? Endoparasite is a parasite who lived in the internal tissue or organ of its host, such as worms, cacing. But unfortunately, this parasite love to live together with us humans or some of these domestic or wild animals, and we are actually the preferred host ever for this parasite to live happily ever after. So believe it or not that we are actually surrounded with this all tiny creature called parasite 24/7 without we realizing it. But for me, the most dangerous parasite ever in the whole entire world is actually human act like a parasite, where they love to take advantage of others. So be extra careful of this human egg parasite. Okay, just joke. But yeah, somehow we need to be extra careful with these types of human being. And for your information, parasitic infection is still a global health problem. It is not a problem that you all believe that only happen in a tropical or poor resources country. It could actually happen everywhere and anywhere, including in highly developed countries, such as in the United States, United Kingdom, Australia, Japan, Denmark, Canada, or even France. Only the nature of these parasitic diseases differs from one country to another country. I will give you two examples to clearly show that this parasitic infection is still a global health problem. Firstly, approximately 260 million cases of malaria has been reported a year, with a total death of 445,000. Another good example, would be 1.2 billion of humans throughout the world actually being infected with a roundworm called Ascaris lumbricoides, and it causes more than 60,000 deaths annually. And though all these cases were not documented frequently, but Center for Diseases Control and Prevention has reported that toxoplasmosis is actually an infection caused by a parasite Toxoplasma gondii, is a leading cause of death from foodborne illness, especially in the United States. Therefore, as I mentioned to you earlier, it is just only a misconception that this parasitic infection only happen in a tropical area. But the fact is, it could happen everywhere and anywhere. And how about this scenario in our beloved country? Malaysia is actually facing with many challenges caused by these various parasitic infection. And it is due to these two main factors. Firstly, the lack of awareness among this disadvantageous population, such as the orang asli. And secondly, the dependency on the foreign workers, which lead to the influx of immigrants workers to our country. And this actually make me feel so worried, especially the illegal immigrant workers. Because why? Because they might unintentionally bring their parasite together with them to our country without we realizing it. And for your information, there is actually a company known as FOMEMA who's actually appointed by our government to undertake the foreign workers' health screening before they could be given a permit to work in our country. However, this is actually only applicable to those who are legal workers, not illegals. Yeah. So with all these diseases that need to be screened, there are actually one disease that related to parasite, which is malaria. So they need to undergo all this kind of screening, including malaria. And for your information, in the middle of the pandemic COVID-19, there is actually a reported of malaria case which happened last month, yeah? And it has been reported happened at the construction site in Hulu Langat, Selangor, where it was suspected that this malaria infection caused by one of the Indonesian construction workers, and perhaps it might be the illegal one. And way back in 2016, there is also an outbreak of this malaria at the Pos Kemar Hulu Perak, which happened among the Orang Asli population. And a total of 137 of these Orang Asli have been found positive with malaria. And again, yeah, it was suspected caused by the illegal immigrant workers. And these two examples clearly show that, yeah, parasitic infections continues to be the major public health challenge in our country. And up to this date, yeah, there is no available vaccine for most of these parasitic infections. Therefore, our government had played a vital role in order to develop this healthcare infrastructure as well as implementing the point of care diagnostic. So we as a researcher, especially those who are working in the field of parasite, what we can do is continuously educate our society and community, and also keep raising the awareness, especially on the hygienic practices, because this is the only tool to curb the spread of such infection. And this what I have been doing for the past few years, where I actively featured in our local newspaper with the aim to educate our society and also to keep raising the awareness, especially on the hygienic practices, and to make them aware and alert that this parasitic infection actually still exists in our local population. So, for a better management of these parasitic diseases, there is actually need for the early diagnosis. Because if we diagnose the condition earlier, and if we give a better treatment and a proper treatment, obviously, it will be the best way in order to reduce the mortality as well as the morbidity that due to these parasitic diseases. So, ladies and gentlemen, when we talk about diagnosis, yeah, for many years, microscopic has been the only available tool for the detections of parasite through inspections of various human samples ranging from blood to CSF. However, yeah, the diagnostic approach seems to be in a sharp contrast as compared to the other field, such as viral and also bacteria, where most of this innovative modern technology in that particular field, yeah, have replaced most of this classical method for the past 2 decades. So this lack in parasitology diagnostic innovation can be partly explained by the fact that the detection and the differentiations of cysts, ova, and larvae of the parasite, when performed by a well-trained microscopist, are technically simple, fast, and inexpensive. And furthermore, microscopic technique also allow us to detect unexpected parasite, which we may discover coincidentally through the screening process. And it is pretty much helpful when the clinical presentation is atypical. So in Malaysia, right, conventional microscopic technique continues to be the first line diagnostic procedure in most of these clinical parasite labs. For example, highly experienced microscopists will use the light microscope to examine the mounting specimen in a saline or iodine preparation in order to detect the cyst, the trophozoite of the protozoa, as well as to identify the aids of this helminth. And this method was known as direct smear method. It is the most simplest technique, it is the most easiest technique, and it actually involves a very minimal cost. So in order to increase or to improve the detection and also the visualizations of this parasite, we could also integrate some chemical throughout this staining process. All right. And for example, this trichrome staining is one of the method of choice in order to diagnose most of the human intestinal protozoa from the human stool samples. Besides this trichrome staining, we could also use Ziehl-Neelsen, and this staining is very useful in order to identify the oocysts of this Cryptosporidium. Besides that, there is also one staining technique known as gram chromatopignon staining, which is, for me, is the best method so far in order to identify the spore of the Microsporidia. And likewise, Gimster staining can provide us a rapid method for the detections of species such as Plasmodium and also Microfilariae in a thick as well as thin blood film preparation. Therefore, why this conventional method still being used or still being the preferred method of choice, especially in economically disadvantaged region? Why? The one and only answer for this question, because all this microscopic technique actually involve with a very minimal cost. And for the past few years, there are actually a lot of questions, a lot of issues has been debated among the researchers due to this continuous use of this microscopic method in the diagnostic lab. And if you'd like to ask me, as a first-hand user of this method for many years, despite of its low cost, I need to admit that by using this microscopic method, it's actually facing with a lot of limitation as well as challenges. For example, by using this technique, it actually requires us to have a high level of skill as well as knowledge. Why I say so? Because throughout this conventional method, you are only able to identify the morphology of this parasite. So that means you need to have a very good knowledge about this parasite, because all this parasite is actually in a small size ranging from 2 micron to 10 micron. So it's very small creatures. Furthermore, by using this microscopic technique, a lot of researchers have published their works and prove that this microscopic method is actually characterized by a low sensitivity and specificity, ranging approximately 40 to 50%. And by using this microscopic method, usually we need more than one sample per patient. Why we need more than one sample? Approximately, we need 3 samples per patient. Why? Because it is due to the intermittent natures of this parasite shedding. It means somehow hari ini dia ada, kita boleh jumpa, besok kita tak boleh jumpa, lusa kita boleh jumpa. So that's why in order to confirm the diagnosis, it is possible for us to take at least 3 samples per patient. So increased labor costs. So in what terms that it could increase labor costs? So if you still remember that previously I told you that by using this microscopic method, the most important thing that is you are skillful enough, yeah, to do all this smears technique, yeah, to do all this staining, as well as you also need to have a very strong knowledge about this parasite. So in order to maintain the level of the skill as well as the knowledge, we need to send our technologies, we need to send our microscopies for this continuous course, yeah, in order to maintain that. So indirectly, it will also increase the labor cost. Okay, in case of this reading slide, somehow we also need more than one readers in order to confirm whether it is a true positive or true negative, especially in the case of the infection is not really frequent or not commonly found in that population. So somehow we need 2 or 3 independent reader, and indirectly it will cost this microscopic technique as a labor and time intensive. And finally, the biggest or the major limitation ever for this microscopic technique is that it unable to distinguish between the pathogenic as well as the commensal parasite species. For example, in the case of Entamoeba histolytica, and I will be further explain about this Entamoeba histolytica on my next slide. So stay tuned. So, despite the limitation, yeah, we need to admit that all major intestinal protozoan, as well as helminth infection, are still solely dependent on this microscopy for diagnosis. We need to admit that, and we cannot ignore or reject the microscopic technique because till now, yeah, we still use it, and some of the diagnostic lab make it as a gold standard in screening the parasitic infection. So therefore, for the past few years, many researchers, including our research team, have tried our best to do some improvement and enhancement to the current method in order to increase their sensitivity, specificity, as well as their reproducibility. So these are the some examples throughout the conventional method, but it has been doing some improvement and enhancement. All right, the first study was conducted with the aim to evaluate the wintergreen oil in comparison with xylene in weekly trichrome staining. And as I mentioned to you earlier, this weekly trichrome staining is the gold standard method ever in order to diagnose most of these human intestinal protozoa from stool samples. Yeah, and one of its process actually include this xylene, which xylene will act as a curing agent. As we know that xylene is actually a toxic, a carcinogenic, as well as a fire hazard chemical. So the long exposures towards this xylene actually can lead us to have a cancer. So with that justification, yeah, with that strong justification, so it actually gives us an idea on actually it is an urgent need to replace the xylene with other chemical. And in this case, we are using the wintergreen oil, or known as a methyl salicylate. So by using 500 stool samples, and we did it triplicate, yeah, we found out that no additional distortion or shrinkage of protozoa was observed. So actually, it is a good sign, yeah? So the second finding showed us that these protozoa of interest appeared more deeply stained as compared when we soak or when we clear the slide using xylene. Another good result of findings. And finally, when we observe the slide under light microscope, we found out that actually it gives us a better contrast, which means it helps us in order to visualize the parasite even more clearly. So why this thing can happen? It's actually due to this high refractive index of this wintergreen, which give the value of 1.53. So it means that the higher the refractive index, the better contrast of the background it will give to us. So the conclusion for these findings, it recommended us to use this wintergreen oil in order to replace xylene in the procedure of weekly trichrome staining. And I also did get feedback from other researchers. They told me that actually wintergreen oil, despite of being using in the field of parasite, this wintergreen oil can also be used in a histology as well as the entomology field, where they could mount the mosquito larva by using this wintergreen oil. So, my dear fellow friends, if you are actually using this xylene in one of your lab procedure, I would highly recommend you to change it to wintergreen oil, because it is quite cheap, and most importantly, it is not toxic at all. So the second study actually was conducted with the aim to develop the concentration procedure that would improve the sensitivity as well as the specificity for Cryptosporidium oocyst detection in human stool samples. Again, by using 500 stool samples, did it replicate? We found that by using this modified formalin ether concentration, what are the modification? Later I will tell you what are the modification. It's actually involved with a few steps of centrifugation and also few steps of sieve. Kita tapis, kita turaskan guna gauze, by using gauze. So we found out that initially these samples cannot be detected under one high power field of microscope. But when we use these modified techniques of formalin ether, it gives us a vice versa findings. So this modified formalin ether is pretty much important and useful for us, especially in order to detect with light infection. So the conclusion for this finding, again, it is a recommended method for you to use, to replace this conventional method with this modified formalin ether technique. And I think it is a good for us to really look into it, as the cases of cryptosporidiosis is keep increasing currently due to this immunocompromised patient such as HIV patient. And the third study was conducted with the aim to evaluate the staining technique which we developed in-house, known as gram chromatograph Kenyon staining, GCK, with a comparison with the conventional method known as Weber modified trichrome technique. And as I said, this GCK staining for me is one of the best method for now, because it really helped us to identify the small size of this microsporidia spore. And this microsporidia spore actually range between 1 to 4 micron. So it's very small in size. And besides that, inside the stool samples, inside our human stool samples, besides we have this parasite, bacteria, we also could found the fungal spores. So fungal spore and this microsporidia spores somehow will looks alike. So that's why we really need to use a very good staining in order to pop out this microsporidia spore. So by integrating with the gram and also Kenyon staining, we found out that it would actually increase the color intensity of the desired spore, in this case, microsporidia spores. And another fantastic finding ever by using this GCK, it could actually stain the polar tube of this microsporidia spore in dark blue color. So this polar tube is a typical and unique characteristic for this microsporidia spore. So as you can see here in this picture, there are actually, it looks like this spore is wearing a seat belt, where the seat belt has been stained in a dark blue in color. So that is actually the polar tube. And as you can see, it really helped us, in identify this microsporidia spore. So when we compare the sensitivity, the specificity, as well as the kappa value, it totally gives us a very high and a very good in agreement, which means that this developed technique, in-house developed technique, can be used as a replacement for this Weber modified trichrome staining. So in terms of time that need in order to complete this procedure, it clearly showed that by using this GCK staining, it gives us approximately 40 minutes, to complete the procedures and allow us to observe under light microscope. Whereby, if we use this Weber modified trichrome technique, it gives us approximately 2 hours. So better you use this GCK. So in conclusion for this study, it clearly recommend that staining technique, this GCK staining technique, has a high validity in the detections of microsporidia spore in a human stool samples. And again, with the increasing of these immunosuppressive, immunocompromised patient in our country, it's also indirectly give the rise of this microsporidiosis. Because microsporidia as well as cryptosporidia is known as the opportunistic parasite. Dia mengambil kesempatan, especially to those who have this low immune system. So currently, there are a lot of cases reported among HIV patients where they actually develop into this chronic diarrhea due to this microsporidia. So I think it is a best time for us now to implement this GCK staining, right? So if you want to know more about the techniques that I have been explained to you just now, or any other techniques that have been made some improvement and enhancement, I would like to suggest you to buy this book, please. To buy this book, because you will, I will guarantee you that you will satisfy. Because the procedure is very specific, and some of this procedure that published in this book has been actually published, which well accepted. So because it just costs you only 25 ringgit, So ilmu mengatasi segalanya. So you can find this book online as well. So with all the improvement, and enhancement that have been made for this conventional method, especially the microscopic technique, you might ask me, why we still need to implement the new and advanced method in order just to diagnose this parasite? My simple and humble question to that question would be, it is because science keep changing. Same goes to this parasite. They keep changing in terms of their acid nuclei, right? So I'll give you an example to make you even clearer, right? During my undergraduate studies, way back in 2002, in University of Malaya, right? I was told that amoebiasis is an infection caused by this pathogenic parasite known as Entamoeba histolytica, right? And by that, approximately 10% of the human population actually used to get infected with this Entamoeba histolytica. And out of that, 10% of them usually will be developed into invasive disease, such as the amoebic dysentery. And approximately around 1% will further develop into this extra-intestinal disease, where the example of amoebic liver abscess. But surprisingly, 90% of the cases which infected with the Entamoeba histolytica, the pathogenic strain, could give rise to self-limiting diarrhea as well as asymptomatic infection. And the question here, how come this pathogenic strain, which we know that it could give rise to a disease such as amoebiasis, actually can give to this asymptomatic? Macam tak logik, kan? Asymptomatic maksudnya dia tidak menunjukkan apa-apa kesan, tidak ada apa-apa penyakit, tetapi puncanya is actually caused by this pathogenic Entamoeba histolytica. And after many years, this hypothesis, this concept has been proved that we cannot, sorry to say, we cannot accept this concept anymore. Why? With the descriptions of a new species known as Entamoeba dispa. Okay, it clearly showed that the amoeba, initially Entamoeba histolytica, that we observe under light microscopy is not really the Entamoeba histolytica species. It might be the twin species known as Entamoeba dispa. However, this Entamoeba dispa is the non-pathogenic species. Why I say it is twins? Because under microscopic examination, under light microscope, the morphology of the Entamoeba histolytica and Entamoeba dispa exactly the same. The size, the number of this nucleus, the locations of this charosome, the chromatoid body, everything is almost similar in morphology. So we totally cannot differentiate between Entamoeba histolytica with its twin, identical twin, the non-pathogenic Entamoeba dispa. And it clearly proved throughout the studies, that 90% of the cases which initially claimed caused by this Entamoeba histolytica, when we further investigate, it is actually not caused by the Entamoeba histolytica, but Entamoeba dispa, the non-pathogenic species. But unfortunately, again, we cannot differentiate them under the light microscope. What we can do is by using the more advanced methods such as this PCR, because it will specifically detect the nucleic acid of the parasite. So this concept also has been proved in our study, where initially we found that 18.6% of this orang asli population initially being diagnosed or being infected with Entamoeba histolytica by using the light microscope. Okay, but when we further investigate these cases by using the molecular methods such as multiplex PCR, it really gives us a vice versa results. And actually only 3.2% of this orang asli being found to be infected and positive with Entamoeba histolytica. But initially, we found at 18.6 but by using this multiplex PCR, it gives us that only 3.2% of them infected with Entamoeba histolytica, whereas the remaining of the cases is actually infected with the non-pathogenic Entamoeba dispa, which is 13.4%. And actually, this finding is pretty much important in terms of improving the patient management, so that we couldn't give unnecessary treatment for those who are infected with this non-pathogenic, and at the same time it will also assisting the public health by allowing for the proper infection control measure. So in other words, what I could say that by using this light microscopy in order to identify the Entamoeba histolytica, somehow it could lead us to over-diagnosis, right? So subsequently, it will give rise to unnecessary treatment, right? So benda-benda tu bersangkut paut, right? So, right. So the molecular method, okay, with the widest variety and application, especially in the field of parasitology, is known as polymerase chain reaction, or in short, PCR. So this is the widest molecular technique that been ever used in the field of parasitology. So, ladies and gentlemen, what are actually the basic principles of this PCR? So PCR is actually a technique that used to amplify a segment of DNA of interest. In this case, we would like to search for the parasite DNA. It's not the patient parasite, This is the parasite DNA. And subsequently, by using this PCR, it will produce a lot of copy. In other words, from one single copy to a billions of copies of the uh, targeted DNA sequences. And furthermore, yeah, this PCR will also mimic what actually happens in the cells when DNA is replicated. But for this time, it actually need to be carried in a controlled condition, yeah, in a proper setup, as well as need to use the high-end equipment such as thermal cycler. So the test tube that containing the DNA mixture of interest. Later on, I will explain to you what are actually the DNA mixture of interest. And all these DNA mixture of interest are put together in a tube known as PCR tube. And then this tube will be transferred to a machine, the high-tech machine known as thermal cycler. And finally, we need to set up a few temperature, such as denaturation temperature, annealing temperature, as well as extension temperature. So on my right hand side, yeah, this is actually what I meant by DNA mixture of interest. Okay, what are the recipes that we need to prepare of this DNA mixture? Okay, first we need to have this target DNA. Where could we find this target DNA? Okay, this target DNA is the DNA that you initially extract from the samples. Either you're using this commercial available extraction kits, or you can do it with a conventional method. So this target DNA, also, okay, that is the first ingredient that we need. Then we also need to have primers. Primers is actually the one that we design. And the functions of the primers, it will specifically bind to this target DNA. Right? Beside primers, we also need to have nucleotide, DNA polymerase, buffer, as well as magnesium chloride. So all these reagents need to be prepared in a final volume, either 25 microliter or 50 microliters. It's actually up to you, right? And again, they will mix together, put in a PCR tube, and finally this PCR tube will be transferred to a thermal cycler machine. And again, yeah, do not forget to set the temperature according to the denaturation temperature, annealing temperature, and extension temperature. A quick tips on how to ensure the successful of this PCR reaction. There are actually three important things that you need to pay attention. First, the quality of your extracted DNA. You need to quantify your DNA, whether it is sufficient enough to proceed with the PCR, to proceed with the replications or not. Because low quality of DNA will definitely affect your final outcome of this PCR reaction. Right? So that is the first one. The second one is the primer that you design in-house, or you use it, the one that has been established, developed by other researchers. You really need to ensure that these primers are very, very specific. Okay, these primers and DNA is just like a concept kunci dengan mangga. Okay, kita perlukan kunci yang betul-betul sesuai kan untuk membuka mangga tu. So that concept actually could be applied in this case. Yeah, primers must be specifically designed to specifically bind to this targeted DNA. And the last one is actually the annealing temperature. Annealing temperature, you need to optimize the annealing temperature because different primers, different targeted DNA need to have a different set of annealing temperature. So these are the three things that you really need to put into consideration in order to ensure the successful of this PCR reaction. So apart from this conventional PCR, there are actually other types of PCR which has been frequently used in the clinical lab diagnostic, such as the what we call multiplex PCR, nested PCR, and also real-time PCR. So which PCR that we need to use? Actually, it's up to you. Tepuk dada, tanya selera. Right? So now we go in general about each PCR, so that it gives you an idea which PCR that really suit in your experimental design. So conventional PCR is the most common PCR ever being used in the lab diagnostic. So what is this conventional PCR mean? It means that throughout the process, ya, dalam tube tadi tu, you only need to include one set of primers. Okay, one set, satu pasang saja. And this set of primers actually consists of reverse and forward primers. Okay, so that means bila kita include one set of primers, so it only give us to detect one parasite of interest. So that means after the 2 hours process, yeah, so you can only able to detect and identify only one parasite, one single parasite. However, for this multiplex PCR, it is more convenient to use, as I'm also using this multiplex PCR in order to detect this Entamoeba histolytica and Entamoeba dispa. So what are the basic principles of this multiplex PCR? It actually allows us to include a multiple primers in that single tubes. Okay, because previously kita hanya letak one set of primers, but for this multiplex, you are able to include as many primers as you want. For example, if you put 3 set of primers, that means after the running process, you are able to detect 3 different parasite of interest. So after 2 hours, you boleh detect terus 3 simultaneously or concurrently detection. So menjimatkan masa, right? But again, the successful of this multiplex PCR definitely depends on the primer design as well as the annealing temperature. Okay. Then we have nested PCR. So apa pula nested PCR ni? Basically, nested PCR, the concept is actually same with this conventional PCR. It's just different in terms of you need to run the PCR twice. Okay, conventional you buat sekali, but for this nested PCR you need to run it twice by using a different set of primers. Okay, first run kita gunakan set primer yang pertama. Second run kita guna set primer yang kedua. Kenapa kita perlukan 2 set primer? Because the second set of primers actually will help us to bind specifically at the target size. Right? So that means kalau conventional PCR tadi it just took about 2 hours, so by using this nested PCR, it will double up the time. For example, 4 hours. So ambil sedikit masa lah kan, it's time consuming. And finally, the real-time PCR. So this is the most used PCR technique ever currently. Because why? Because of its advantages. First, it could help us to detect parasite. Okay, sama je dengan PCR yang lain. But the second thing is, it help us to quantify the parasite load. Jadi kita boleh tahu berapa banyak parasite yang ada. So apa pentingnya? What is the importance or significance of us to know up to that stage? Maksudnya kita nak tahu sampai jumlah bilangan parasite. Okay, this actually can be applied, usually can be applied in terms of malarial infection. Okay, where somehow we really need to know the parasite load, the parasitemia value in the patient blood in order to ensure the successful of the treatment. Contohnya pada hari pertama, when we give drug A, kita tengok the parasite load tu, contohnya it start with 10,000. The second day, kita tengok it dah decrease to 9,000. And then following 8,000, 7,000, 6,000, 2,000. So it really helped us to monitoring the successful of this treatment. So that's why currently the real-time PCR is the method of choice. Yeah. And besides that, by using this real time, it also help us to eliminate the cross-contamination. Okay, cross-contamination ni selalu berlaku. Why? Because this PCR method is way too sensitive, sangat-sangat sensitive. Takutnya nanti during the preparation, the pre-PCR preparation, takut masa you pipette, you prepare all the secret recipe, takutnya you bercakap ataupun terbatuk. Okay, all those things actually contain DNA. Takut-takutnya nanti DNA, the unnecessary DNA will also be included in that PCR, and subsequently, you also might find other DNA beside the DNA of interest. So by using this real time, it can eliminate all this cross-contamination, right? So, when we talk about PCR, for sure, with all these orang kata benefit dia, okay, memang banyak. There are a lot of advantages of using this molecular method. However, I only will highlight the 4 most significant ever by using this PCR method. First, the advantage of using this PCR, definitely, it will help us to improve the sensitivity for diagnosis. Okay, for example, the detection limit of thick blood smear is about 50 parasite per microliter of blood. Bermaksud when you use the conventional method one, which is the BFMP staining and observe under microscope, you could only able to detect whether it is positive or not if, let's say, it contained 50 parasite per microliter of blood. But by using this PCR, it can achieve a detection limit of even lesser than 5 parasite per microliter of blood. Bermaksud dia kalau dia sedikit pun kehadiran parasite ni di dalam darah, dia masih lagi boleh didetect by using this PCR. So itu bermaksud how sensitive this PCR method, right? And the high level of sensitivity is not only allow us for the detections of low level of parasitemia for clinical diagnosis, per se, but it is also potentially useful in a screening of blood products by blood bank. And actually, when you go and derma darah, actually the blood products need to be screened. You need to ensure that your blood is free from any infectious diseases. So in order to do that, it will be good if you screen the blood products by using this PCR method, right? And then the second advantages of using this PCR is actually help us to precisely differentiate the parasite species. Ini yang penting dia, orang kata the gist ni. Kenapa kita perlu gunakan PCR? So, exact parasitic speciation is sometimes not essential for therapeutic purposes. Okay, that one we need to admit. Macam ni, kalau patient tu pergi ke hospital dan didapati positif dengan parasite, definitely the patient need to be treated. Tak kisahlah dia spesies apa, sebab nama pun parasite, especially if the patient infected with the pathogenic parasite and give rise to the clinical symptom. Okay, for sure doctor will give treatment in order to treat that particular patient. Okay, so tak perlu kita nak tahu spesies apa. But kenapa kita perlu tahu up to species level? Okay. However, in most of these epidemiological study, as well as in a public health, it always desirable to have or to identify up to species level. Why? Because some of the morphologically similar parasite may have a different pathogenicity, may have a different modes of transmission, or they might even have a different intermediate host, which all of this will give an impact towards the control programs. Okay, in parasite especially, besides we treated the patient, okay, we also really need to know the cause of the problems or the roots of the infection. Kenapa kita nak perlu tahu sampai ke tahap-tahap level itu? Because we would like to cut the transmission modes or break the infection chain. Okay, macam Datuk Dr. Syam cakap kan, kita nak putuskan rantaian supaya penyakit itu tidak terus menerus merebak di dalam masyarakat. So that concept is actually can be 100% applied to this parasite. Okay, orang sakit kita treat, but we actually need to know what are the cause of that particular patient. What are the cause? Why? Where he get the infection? In what source? So that's why in the epi and public health, we really need to know up to the species level. For example, macam tadilah Entamoeba histolytica, betul tak? Kalau kita tak go up to species level, kita akan terus menerus give unnecessary treatment. Okay, so bila kita putuskan rantaian, when we know the roots of the transmission, akar punca-punca dia, then InsyaAllah, jangkitan ini tidak akan terus berada di mana masyarakat. I just give you a good example lah. Up to this date, the prevalence of soil-transmitted helminths, especially trichuriasis, is still highly prevalent. 90%, more than 90%. It means that kalau you sample 100 orang, ya, 90 orang adalah positif dengan jangkitan cacing. Hello, kita dah masuk tahun 2020. Apa ni cacing-cacing ni? Kalau tahun 60-an, bolehlah nak ada jangkitan cacing. But now, hello, semua orang pakai kasut. Itu apa yang kita nampak. If we go beyond that, beyond our population, beyond our community, there are actually still exist human who still not wearing a proper shoes or slippers. So that's why, walaupun community tu kecil lah. Okay, I just give you example orang asli, because my main concern, my main study subject is actually orang asli. So kita tak boleh ignore mereka because they are actually also a part of our society and community. So why should we ignore and abandon them? Walaupun kita tak apa orang asli dia duduk kat hutan, but no, they're still a human being. We still need to take care of their health, right? So that's why we really need to know up to that species level. So another good example would be mixed infection. Okay, mixed infection usually happen in the terms of a malarial infection, in the case of malaria infection. Okay, for your information, ladies and gentlemen, malaria is actually caused by 4 species lah. Okay, sekarang ni saya cakap 4. Later on, you will know that actually not 4 anymore. We already have 5 human malaria. Okay, sekarang ni konsep dia 4. Okay, somehow we could get the single infections of malaria, katalah dengan species A. But it is also somehow could be reported that we might infected with a mixed infections of malaria. Malaria species A dengan malaria species B. And why we need to know whether it is a single infection or mixed infection? Because the anti-malaria that will be given is totally different, right? So, fail to recognize this mixed infection may lead to the treatment failure. Alright, sepatutnya orang tu boleh cepat sembuh, but subsequently maybe dia akan meninggal because setiap spesies malaria ni it give a different range of clinical manifestation. Okay, and the time for this species to replicate also different. Ada yang cepat, ada yang lambat sedikit, ada yang lambat banyak. So we really need to know the species of this malaria. Okay. So the third example, by using this molecular method, it also helps us to identify the target genes of anti-malaria as well as their mutation gene. Okay, kita perlu tahulah, kita tak nak nanti drug yang kita bagi tu dah resistant. So by using this molecular, it actually helps us a lot in identify the target genes as well as the mutation genes. And the ability to detect this anti-malaria resistance is of a great importance, both in therapeutic agent as well as in a global surveillance resistance. And finally, the use of the PCR method is pretty much important in order to screen those who are at a higher risk. Okay, for example, in the case of transfusion-transmitted infection, the use of this PCR may potentially be beneficial in the prevention and management of the transplant-related parasitic infection, such as malaria, toxoplasmosis, and strongyloidiasis. So these are the 4 major significant advantages of using this PCR method. And now, the most interesting part, tadi kita dah cerita pasal basic principle, kita dah cakap jenis-jenis PCR. Now we would like to see the real applications of PCR in epic study as well as diagnosis of parasitic infection. Okay. Okay, the first one, as I mentioned to you just now, that by using this multiplex PCR, it actually helped us, our team, to identify between the pathogenic Entamoeba histolytica and also the non-pathogenic Entamoeba dispa. Okay, itu satu case. But by using the same method, multiplex PCR, surprisingly, it also helped us to be the first research group ever in Malaysia to isolate the third species of Entamoeba Moshkovsky. Apalah pula Entamoeba Moshkovsky ni datang daripada mana? Alright, okay, cerita dia macam ni. Okay, tadi kita tahu Entamoeba histolytica have its twin, which known as Entamoeba dispa. But this Entamoeba dispa is the non-pathogenic one. Okay, tak perlu takut. If let's say you are being diagnosed having this Entamoeba dispa, no treatment is needed. Okay, so when we actually managed to isolate the third species, which is known as Entamoeba Moshkovsky, surprisingly, this Entamoeba Moshkovsky also give a similar morphology with the Entamoeba histo and Entamoeba dispar under the light microscope. So that means ya, kalau you lihat initially you katakan this is the Entamoeba histolytica, but actually you could ask yourself, you need to ask yourself. Now, from what I have learned throughout this Dr. Tengku presentation, I cannot really release this result. I need to further confirm by using molecular technique, because the one that you see under microscopic, such as Entamoeba histolytica, currently is not only Entamoeba histolytica. It could be Entamoeba dispar or Entamoeba Moshkovsky. Okay, now we need to know whether this Entamoeba Moshkovsky is really a pathogenic species or not. Kalau dia tak pathogenic, no treatment is necessary, right? So, okay. Throughout the studies that have been reported from many researchers around the world, initially, it is thought that this Entamoeba Moshkovsky is a non-pathogenic species. But however, currently, this species has now been associated with a diarrheal diseases. That means sesiapa yang get infected with this Entamoeba Moshkovsky, actually it could give rise to the clinical symptoms, signs and symptoms such as diarrhea. So, sekarang ni kita nak classify this E. Moshkovsky ni as a pathogenic or non-pathogenic. Kita tak tahu, right? So, in our study, okay, as we managed to isolate this Entamoeba Moshkovsky in 13 orang asli, 13 orang asli. Out of these 13, we found out that 3 of them actually give rise to this clinical manifestation. And these 3 persons actually developed into diarrhea, watery diarrhea, abdominal pain, as well as fever. Menjadi satu tanda tanya whether this E. Moshkovsky is actually a real pathogenic species or actually it is a non-pathogenic species. Kita tak boleh nak buat satu konklusi. Because why? Because these 13 orang asli, for your information, they are actually infected by a single infections of E. Moshkovsky. There is no co-infection with other pathogen protozoa ke, co-infection with helminth ke, no. This 13 orang asli is actually infected with a single species of E. Moshkovsky. And this actually give us some ideas, macam dia punya pathogenicity ni boleh diragukan, doubt. So actually the true pathogenicity of this E. Moshkovsky needs a further confirmation through in vivo studies. So sesiapa yang nak buat kajian tentang this E. Moshkovsky, now you can use this as a stepping stone. You need to go into in vivo study in order to know whether this E. Moshkovsky is a real pathogenic or not, right? So, okay, itu pertamanya lah, okay, kegunaan multiplex study. Okay, another things, besides we use this PCR to differentiate the species, actually we also need to look within the species itself. Okay, their different strain, their genetic diversity. Kita ambil contoh macam ni lah. Entamoeba histolytica, for many years, it has been known that this parasite is the pathogenic species, Entamoeba histolytica, no doubt. Okay, however, the clinical outcomes or the clinical presentations of this Entamoeba histolytica is likely due to be influenced by the different strain of the parasite that exists within themselves. Okay, sebab itulah somehow we could find a patient who infected with Entamoeba histolytica, somehow they will present with a severe, the moderate or more chronic infection. Okay, because this Entamoeba histolytica could give rise to a watery stool, okay, bilik, diarrhea, ataupun lebih teruk sikit, it can give rise to amoebic dysentery, berak berdarah, and dalam paling teruk lagi, it will cause you amoebic liver abscess. Nampak tak di situ? Dia ada perbezaan dia punya kepathogenicity kan. Okay, a different pathogenicity. Tetapi punca dia sama, Entamoeba histolytica. What actually give rise to this various clinical manifestation? Actually, yang give rise to this clinical manifestation is actually the strain that exists within their species. So for example, the invasive strain of this E. histo actually can cause ulcer of your intestinal epithelium and subsequently can develop further into amoebic liver abscess. So what is so important to know the genetic diversity that could exist within the species? This would be a major impact on the subsequent course of treatment that given to the patient, right? So another examples, yeah, in a real application. So we use this PCR in order to diagnose a patient with giardiasis. Okay, giardiasis ni penyakit yang disebabkan oleh Giardia intestinalis. So this Giardia intestinalis definitely a pathogenic parasite. Dia tak macam Entamoeba histolytica tadi yang dia ada kembar-kembar dia. Okay, kembar tak bahaya, kembar separa bahaya. For this Giardia intestinalis, it's actually 100% pathogenic. So those who are infected with this Giardia intestinalis definitely need to be treated. Okay, tapi persoalannya di sekarang okay, kenapa? Why we need to look into the molecular level, up to molecular level? Because okay, when we study up to molecular level, it gives us an idea that actually this Giardia consists of different strain. Okay, they consist of 8 different assemblage. Dalam arti kata 8 different strain. They have assemblage A up to assemblage H. So why we need to know up to this assemblage? Lantaklah dia strain apa pun, because at the end we still need to give this patient treatment, right? Okay, itu dari segi perubatan lah. Okay, doktor akan terus merawat, tetapi dari segi ilmu public health, again, we need to know each strain because this Giardia intestinalis is actually a host specific distribution. Maksudnya, setiap strain itu mempunyai perumah yang spesifik. You might ask me, why we need to know up to that host? No need, just give a treatment. Okay, again, I would like to stress, why we need to know up to the host level? Because we really need to cut the mode of transmission. Kita nak putuskan rantaian. Kalau tidak, after a month, that particular patient will come back and, penyakit yang sama, memberi rawatan yang sama. So benda itu berulang. It's just like a merry-go-round. Betul tak? So we really need to know up to what is actually the source of this infection, because this Giardia is a host specific. Okay, for example, assemblage A, Giardia assemblage A is known to be host specific to this animal. Maksudnya dalam animals ni banyak Giardia assemblage A, particularly dogs and cats. Whereby the Giardia assemblage B is known to be actually a human strain, because banyak kan yang kita jumpa adalah dalam manusia yang kalau assemblage B. Okay, when we use molecular, we can identify and we can detect up to this level tau, assemblage A, assemblage B, assemblage C, D, E, up to H. So in our study, we found that 10.2% of orang asli infected with this Giardia assemblage A, whereby only 5.9% of them infected with Giardia assemblage B. So again dua-dua kumpulan ni kalau pergi ke hospital, for sure they will be given the same treatment, the same drug. Okay, sama, Giardia lah. Okay, but macam tadi kan, our concern is to cut the transmission route. So kita tengok because it is a host specific. Okay, when we do the risk factor assessment or risk factor analysis towards each of the patient, we found out that those orang asli that infected with assemblage A are actually, they have a very close contact of their household pet, such as dogs and cat. Dan bila kita nak relate balik dengan fakta yang this is a host specific distribution, yes, assemblage A actually can be found abundant in animal such as dogs and cat. And when we look into their daily lifestyle, they actually sleep together, eat together. Mandi, saya tak tahulah mandi dengan anjing ke tak, saya tak tahu. Okay, I reserve my comment. But what I could see actually, they actually eat together with this dog. They mingle around. They just treat this dog as a part of their family members. So that's why they could have a high risk to get this Giardia assemblage A. Whereby for those who are getting this Giardia assemblage B, when we do some these risk factors analysis, we found that actually they get infected if one of their family members harbor this Giardia. That means human to human transmission. Okay, dalam kes ini kalau kita tengok cara dia mendapat jangkitan ada dua cara. Kalau SMHA, for sure dia mendapat jangkitan itu menerusi hubungan yang rapat dengan animals. Manakala mereka yang mendapat jangkitan B, Giardia SMHB, mereka ini adalah daripada human to human strain. Okay, apa yang kita boleh lakukan as a public health? Okay, dalam public health field. Okay, macam tadi kita cakap kita nak putuskan rantaian kan. So what we can initially do is actually we need to find the source. In this case, we also isolate this dog and cat stools samples. Why we isolate that? Because we would like to see whether this dog and cat, they also harbor Giardia intestinalis and what strain that this domestic animal harbored. Through our findings, it showed that this dog and cat also harbored the same strain as their owners. So that means tuan dia mendapat jangkitan bila ada hubungan yang rapat dengan haiwan ini. And you need to know that this Giardia is a zoonotic strain. Okay, dalam parasite ni, walaupun orang anggap dia neglected, orang tak kisah, they just pay attention more towards the viral and as well as bacteria. But you need to know that most of the parasitic infection currently is actually a zoonotic strain or zoonotic disease, which means it's originate from the animals, but after sometimes due to these mutations or whatever, these animal strains can now be transferred to human. Macam lah tu orang kata apa H1N1 tu semua kan, asal daripada burung lah, ada yang daripada babi lah, dan sebab pakai babi, kinzi, right? So it same goes to this parasite. Now our concern because it is a zoonotic strain. Besides zoonotic strain, some of the parasite also can cause a waterborne. Okay, maksud dia boleh berada di dalam air. Itu bahayanya parasite that people tend to neglect. Macam tak kisah, tak pedulikan kan, tak apalah. Okay, so sekarang those yang actually watching this webinar session get the idea how important parasitic infection will be. Alright. So, okay, this is actually the current research which currently being conducted in I Promise Lab. Okay, one of my PhD student, Rosnani Hanim, is currently conducted this study, which is Akantamiba. Okay, what is Akantamiba? Akantamiba is a free-living amoeba where you can find it abundantly in the environment, such as in a beach water, in a hot spring water, in a soil, as well as in a contact lens, perhaps your contact lens. Okay, so because it can be found abundant in the environment, and dekat sini kita punya concern because it also can be found and isolated from your contact lens, including your contact lens paraphernalia, contact lens cases, contact lens solution. Okay, so we really need to know whether this parasite known as Akantamiba, whether they have a pathogenic strain or not. Sebab kita terdedah dengan mereka 24/7 actually. So we need to study up to their molecular level whether does this pathogenic strain exist or not. Kalau tak ada, tak apalah, bermaksud dia tidak berupaya untuk menyebabkan penyakit. But if let's say there are one pathogenic strain, then we need to be more extra careful because they can be found abundantly in the environment. And through our study, by using this 18S RNA gene target, we could found that this Akantamiba can be further differentiate up to 21 genotypes. Ada 21 strain. Okay, 21 genotype, T1 until to T21. And we found out, okay, we managed to isolate this Akantamiba from beach water. Kita adalah pergi ke beberapa kawasan air laut, kita tak boleh cakap lah, nanti orang marah, nanti tak laku hotel tu. And then kita juga pergi isolate daripada hot spring water. Hot spring water now be a part of the recreational water tau. Orang pergi beryoga kat dalam air water tu macam dia orang pergi bermeditasi di dalam air kolam air panas tu, dia rendam. Okay, rendam. So that's why we try to isolate whether this Akantamiba can live in this hot spring water or not. Satu lagi, our samples would be the contact lens. Okay, contact lens. So we managed to find out that yes, dekat beach water, dekat hot spring water, there are actually a lot of genotype. Dia ada genotype T3, T4, T5, T6. Ada yang memang specific genotype for that particular beach. Ada yang general genotype. Okay, but most of the genotype that has been successfully isolated from this beach water, as well as hot spring water and contact lens are actually T4 genotype. Paling banyak tambah di dalam contact lens 100% of this Akantamiba that exists is actually the T4 genotype. And based on the previous study, they have identified that more than 90% of Akantamiba keratitis, penyakit cerita mata, were found to be associated with this T4 genotype. Mampus kau. We are actually exposing ourselves with this pathogenic strain of Akantamiba. Alright, so tak hairan lah in the future we could develop into this Akantamiba keratitis or encephalitis. And their pathogenicity also have been tested in our lab. It clearly showed that this T4 is highly pathogenic. Bila kita kata highly pathogenic, once you dapat this strain, okay, you berpotensi untuk develop into this keratitis or even encephalitis. So that's why we need to know up to this molecular level. Kalau tidak, orang akan cakap Akantamiba tak apa, dia free living je, memang dia wujud pun. But we need to know kewujudan dia tu adakah membahayakan kita, mengancam nyawa kita ataupun tidak. Right? So my last example for this application, ini menarik. Right? This is very interesting. Okay. Just now I mentioned to you that we actually can be infected with 4 types of human Plasmodium malaria. Okay, penyakit malaria ni common, korang je yang tak tahu, right? Yang korang tahu dengue, malaria pun common. Okay, but okay, kita ada 4 species lah. Okay, we have Vivax, the most common species that infect our community, Vivax. We have Falciparum. Falciparum ni ada tapi jarang-jarang. Usually Falciparum ni imported cases. Okay, we have Plasmodium malariae, the third species Malariae, and the fourth species Ovale. Ini 4. Basically orang tahu for the past few years, kalau you baca kat textbooks pun, parasite textbook, they only mention these 4 species. Right? Okay. Dengar baik-baik But since 2004, we actually have another species which have a potential to infect human. Okay, and it clearly showed that this the 5th species able to infect human. Nama dia Plasmodium knowlesi. Okay, knowlesi ni bukan satu malaria spesies yang baru wujud daripada langit ketujuh, bukan. Right? This knowlesi is actually a markup malaria. Malaria bagi kera. Okay, but after somehow, malaria dalam kera ni now can be transmitted to human because this knowlesi, again, it is a zoonotic disease. Okay, first cases was reported, if I'm not mistaken, in 2004. The case of malaria keep increasing, especially in Borneo. Nak tahu mana? Okay, dekat Sarawak, dekat Sabah. Kalau Sarawak specifically Kapit. Okay, di Kapit, di Mekah, kawasan-kawasan itu. Sebab kawasan-kawasan itu masih lagi dipenuhi dengan hutan-hujan tropika, right? So banyak kera. Okay, bila ada kera yang ada dis malaria, okay, ada pula vektor dia nyamuk yang bersesuaian, gigit kera itu, dia ambil that parasite, gigit manusia, transfer into manusia. Because this knowlesi is a zoonotic strain, so this strain is capable to infect human as well. So that's why pada waktu itu, okay, yang malangnya, unfortunately during that time 2004, not many people are using this PCR. Actually kan, in parasitology field, dia punya diagnostic method ni semua ni yang conventional method, semua yang jenis lama-lama je. Okay, kalau kita nak compare dengan virus dan bakteria, this particular field usually they are more updated in terms of diagnostic. But in parasite kan semua masih menggunakan this conventional method yang kata alat-alat zaman Tok Nadung gitu kan. So by using this conventional method it is unable to differentiate the species correctly. Ini apa yang berlaku pada tahun 2004. Sehingga kan ada beberapa pesakit yang dah meninggal. Because why? Because initially they diagnosed the wrong species of malaria. By using this microscopic, they diagnose species Malariae. Because this malaria is not common in Malaysia. And another thing, malaria ni, dia mereplikasi di dalam darah, it takes about 72 hours. ambil masa yang lama untuk dia nak membiak secara banyak di dalam tubuh badan manusia. But dia peliknya, bila dia tengok that specific patient, that particular patient, within 24 hours, dia punya jumlah malaria tu makin meningkat dengan banyak. So they get confused tau. By observe under light microscope, it seems looks like malaria species. Tetapi dari segi replication dia, malaria takes about 72 hours. But this patient, every 24 hours, it develop very fast. So dia akan menyebabkan kesakitan yang lebih pantas. So because of this misdiagnosis, so they give a wrong treatment dan sebagainya. So subsequently, this patient died. Okay, ada beberapa orang, you boleh baca kat jurnal pun, dilaporkan pun, ya. So what they did is actually, they send the blood sample to IMR. Because previously IMR is the solely institute that use PCR to identify this malaria. So by using this PCR, they found out that actually, yes, you are actually misdiagnosed. This is not the malaria species. This is a Knowlesi species which originally come from macaque. Okay, again, bila diorang ambil darah macaque tu, kera tu, okay, kera, macaque tu kera lah, bukan monkey tu monyet, ni kera ni macaque. So dia ambil darah macaque tu, kera tu, dia try sama, memang macaque tu ada, dalam darah dia ada this Knowlesi dan strain yang sama. So dari situ kita beritahu, yes, orang ini dapat infection because exposed to this macaque lah. Right, so yeah, with the help of this PCR, it can actually give us a correct detections and correct identifications of parasite. Okay, so itulah betapa pentingnya kita menggunakan this molecular. And currently, ya, dekat Sabah dan Sarawak ni, there is actually one malarial referral centers which based in UNIMAS. Dia dah jadi one of the center of excellence pun dekat UNIMAS tu, under the management of Professor Dr. Balbersing. So he can be said like fathers of this Knowlesi in Malaysia. And yet, for information, yeah, this Plasmodium Knowlesi that give case to malaria, the Borneo case actually stated the top 2 in the world. Dia report lebih kurang beribu-ribu case of Knowlesi. Whereas in Semenanjung juga tinggi case dia, lebih kurang dalam 200-300. And we do have a case dekat Kuala Kubu Baru, bila orang tu pergi memancing, ya, and then ada kera berkeliaran kat situ, ada pula vector dia gigit kera tu, and then gigit pula orang yang memancing tu, dia dapat Knowlesi. Right? So, yeah. So in short, yeah, why should we use, okay, this molecular diagnosis for parasitic diseases? I'm pretty sure that you have all this answer right now, yeah, based on these examples. By using this molecular method, it is actually a powerful tool, a powerful blend of speed, cepat, accuracy, sangat tepat, and also it allow us for these multiplexing capabilities. Dia boleh membantu kita untuk detect lebih daripada satu parasite dalam satu masa. Right? Itu kebagusan by using this PCR. However, dalam bagus-bagus dia pun ada juga beberapa keburukan dia. There are few issues that actually give rise for this molecular technique. First, by using this technique is actually involved in a high cost. Yes, I need to admit that. Just to buy one PCR machine, it costs about 30,000 to 40,000. Okay, so sangat mahal, and you also need to have a very full equipped lab infrastructures, because this PCR involve pre-PCR, dia kena ada bahagian pre-PCR, dia kena ada bahagian post-PCR, so lab setup must be bombastic, right? And therefore, in order to use this PCR, it also require an expertise to handle it. Okay, ini some feedback lah that I get from this lab technician, especially the senior one. The senior lab technician usually love to use this conventional. For example, direct smear, staining, because for them, they already familiarized with all this technique. Okay, but in order for them to learn a new technique, which require a very high skillful technique, yeah, in order to prepare the solution, the mixture, to extract, okay, to pipette. Kalau ada yang Parkinson tu, tangan bergetar-getar ni, you tak sesuai. Jangan sesekali get involved in this molecular study, ya, because somehow you need to pipette in a small volume. 2 microliter, 3 microliter dah lah, dengan mata pun tak nampak, macam pipette dengan tangan getar, mata tak nampak kan. Better jangan. Salah solution, salah mixture, you run PCR after 2 jam, buang je ke dalam tong sampah. Alright, so it really requires an expertise, right? So these outcomes, okay, by using this PCR, the outcome, the findings also somehow affected by variables. And last but not least, some of the major challenges lah by using this PCR is actually it prone to the contamination because it is too sensitive. So ada apa saja DNA dia boleh detect, ada apa saja DNA dia kena detect. So that means you need to really take care of while doing your preparation works, ya. You kena pastikan you duduk dalam tempat yang betul-betul bebas, ya, daripada sembarang contamination dan sebagainya. Alright, so okay, dah habis dapat tu. Alright, so pendek kata, molecular method is actually a very good method. Oi, dah lepas lama dah tak apalah kot. So saya nak kata satu je. Alright, apa-apa pun method yang kita gunakan, the seronok sangat bercakap kan, tak apalah, ini kita boleh escape. Kalau nak nanti kita buat second version, ya. So will all this new technology actually improve the diagnostic? Yes, definitely. It will help us a lot, and it will also lead us to a new capabilities. And perhaps, medical practitioner must be sensitized to utilize all these capabilities. Therefore, they need to work hand in hand, with the scientists in order to choose what actually the best method in order to diagnose that particular disease. Okay, so healthcare provision paradigms need to invest more for the development of newer technologies. So when we use these new and more advanced technologies, therefore, the goal of the right result at the right time, on the right specimen, from the right patient, with the result interpretation, based on the correct reference data and at the right place, should be more achievable. Tidak ada alasan lain. So before I end up my presentation, I would like to share some of this interesting, orang kata, interesting developments, in diagnostic, especially in the field of parasitology field. So in the 19th century, okay, people actually use this microscopic examination in order to identify the parasite. But however, in 20th century, people really want more and more simple and cost-effective tools, which involve serology. But currently in 21st century, we are slowly but surely try to shift to genomics, proteomics, and rapid imaging. So again, the choice is yours, whether you still want to stick with this conventional method or you want to integrate. Okay, screening, you gonna this conventional, but to really identify, you can use this molecular method because you need to use this appropriately. All these molecular techniques actually is very expensive and costly. So again, tepuk dada, tanya selera. Which method is the best? It's up to you. Alright, so thank you so much for your kind attention. Dah lebih dah 20 minit kan? Alright, thank you.
All right, thank you, Dr. Tengku, for the interesting knowledge sharing. I didn't stop you because I think whoever wants to stay can stay. It's a free platform, right? So I mean, so I think we will open the floor for questions and answer session. Maybe I read first some of the comments. There's a comment from Associate Professor Dr. Megawati, a suggestion for you. Maybe, hello?
Yeah. I'm still.
Okay. So Associate Professor Dr. Megawati said maybe you can write about this in the media, newspaper, especially your intro part. It's good for the public. Said you may write about 800 to 900 words about this in the media, newspaper, especially the intro part. I think there's another comment as well saying the same thing from Hadijah Muhammad Yusof. Mudah difahami, mohon diluas sebarkan melalui lain-lain medium penyampaian pada masyarakat. Maybe you can comment on that?
All right. This is what I have been done for the past few years, without maybe you are not realizing it. I'm actively actually featured in some of our local newspapers, in some of the column, where I wrote in layman terms in order to really educate these peoples about this parasitic infection. Paling common lah, you tahu tak penyakit kaki gajah? Okay, penyakit kaki gajah tu actually disebabkan oleh parasite malaria. So benda-benda macam tu saya ada wawarkan di dalam surat khabar lah pada ketika itu. Tapi sekarang insya-Allah, okay, maybe dengan orang kata permintaan ya daripada mereka yang bukan berlatar belakangkan bidang parasitologi, so why not I will try again to feature in any
Salam Dr. Tengku.
Ya, waalaikumsalam.
Salam Dr. Tengku. Yes, I think your article, what you talk today is very interesting for the public, especially. Now we are talking about the COVID, something related. I think it's best you write.
Thank you so much.
Yeah.
Thank you so much for your support.
Very interesting. Thank you so much.
Welcome. Thank you.
Right. Maybe I read the next question from Rosiana Bujang, asking how to buy the practical guidebook. She missed the info.
Okay, you can actually find it in the online actually. You type je tajuk dia tu, nanti dia akan pop up link dia. Alright, you can order it through online.
Alright. Don't for your item staff, please don't forget to fill in the what do you call that, attendance form, which is in the chat. The link is in the chat box, right? A question from Joseph Alagiaraj Tamburaj. How long does it take to detect the parasites in blood?
Alright, so to answer this question, actually, if you realize that in this parasitic field, actually, we are not so into this serology diagnostic. Why? Because this parasite can mimic to or can make our antibodies confused. So somehow, contohnya, kalau dalam case toxoplasma dan sebagainya, somehow, for the early detection to be detect, somehow it takes about 1 week or 2 weeks. So that's why the preferred method is not the serology, but the preferred method for me is still the molecular one, because somehow for certain infection you can detect the parasite in the blood but not at the early infection. Kita perlu tahu awal-awal lagi tu kalau dia dah sakit kita kena treat dia, tetapi kalau daripada awal kita sukar untuk identify dia through blood, so no point. So as for me, how long does it take? Somehow it will take more than a week for the early infections to be exposed, right?
Okay, thank you. There's another question from Shamsul Bahrain Gulam Ali. Great talk. Do you think we should start to integrate One Health concept in diagnosing cases in Malaysia or in research? There's a lot of pieces of evidence that animal and human parasites, including helminths, bacteria, viruses, may co-infect in one or single host population.
Yes. Thank you. I guess Dr. Shamsul. Yeah. So thank you, Dr. Shamsul Bahrain, for your question. Yes. Because currently, parasite field, they are actually integrate with other animals as well as waterborne. So the concept of One Health is actually currently being applied, and I'm also one of the members of the My Ohun One Health University Network, and we are actually really looking into it. And my previous PhD students also did his PhD by integrating all this One Health in order to identify the blastocystis. So yes, I could agree that this One Health concept is pretty much important and can be further into the research. So hopefully I can work with you, Dr. Shamsul Bahrain.
Right.
Can this Dr. Shamsul Bahrain leave your contact email so that I can contact you? Thank you.
All right. A question from Dr. Ramlan Mustafa. I'm interested in what you said a moment ago. One of the reasons this disease spread is due to illegal immigrants. Have you ever presented this finding to the government? Is the problem of these immigrants is not solved until any time the problem of this disease is not solved?
All right. Thank you, Dr. Ramlan Mustafa. Yes, we did actually present and actually voice up this issue. If I'm not mistaken, way back in 2014 or 15, we have this persidangan meja bulat, with all these parasitologists, the public health expert, together with this representative from the government as well as the FOMEMA. And throughout this discussion, actually, we also suggest to increase the panel of the detection, because currently, kalau you perasan, there's only one parasitic diseases that involve throughout the screening, which is malaria, perhaps they also need to screen for the other diseases such as toxoplasma, strongyloidosis, kan, yang macam jangkitan-jangkitan cacing tu. So yes, we already have discussed, but when to really implement that things, it takes somehow times lah, I guess, unless if we are actually the part of this penggubal undang-undang in the future. So pray for me. Who knows? I'll be one of these at school or menteri akan bring this issue. Right?
Right.
Thank you so much, Roland.
All right. I think there's a lot of thank yous and praise for your presentation in the chat, but I don't see any question there. Maybe I open for 1 minute question from anyone can ask from voice, and then if there's no question, we can end the session. Other question? Is there any question from anyone else? Feel free to ask Dr. Tengku.
No question?
Doctor.
Yeah?
Doctor.
Yeah.
To which newspapers that you usually write?
I'm usually wrote to newspapers such as Utusan, Mingguan, Cosmo, Harian Metro, Berita Harian, all these Malay newspaper.
Oh, I see.
Yeah.
You may try writing the Star,
Okay.
the lifestyle section. Yeah, I always write there, and there is one section called lifestyle section.
All right.
So you can write there. And also in the NST, the education section, it is only 800 words, very short and simple.
Yes, definitely. I would like to find this the reporter contact. Yeah.
You just go to you just buy one newspaper and you see the email written there.
All right.
Just submit to the email.
All right. Thank you so much for your help, Doctor.
Sure. I am from APB.
Oh, APB. All right.
Yes, thank you, Dr. Mega.
Sure.
All right.
Thank you.
Okay, so I think we can end the session now. Thank you everyone for participating in this sharing session, and thank you our speaker, Associate Professor Technologies, Dr. Tengku Shahrul Anuar. Thank you very much. Thank you everyone.
Thank you so much everyone. Stay safe and selamat Hari Raya Aidiladha. Berhati-hati semasa memandu. Take care. Bye bye. Assalamualaikum.